Guys, some questions which could come up. Let's look at some slight predictions and how we do them. Question 1. So this is about enzymes. So when we're thinking about enzymes, we're thinking about the lock and key hypothesis. Enzymes have a very specific shape at their active site, keyword, substrate, that fits perfectly into the active site. If the active site changes shape, which we call denaturing, the activity of the enzyme breaks down because the substrate no longer fits the active site. Okay, so let's see. These are all the things you're thinking of when you're thinking of enzymes. Okay, so enzymes can be described as biological catalysts. What is the role of a catalyst? Well, a catalyst speeds up a reaction. Okay, explain why each enzyme can usually catalyze only one type of reaction in the lock and key model of an enzyme action. Well, this is because if we think about let me make this a bit thinner. If we have an enzyme, the reason why they can only, and let me make this active site very specific, the reason why they can only catalyze one specific type of reaction is that the substrate will fit exactly that active site and only one substrate. So this is the substrate and this is the active site. Okay, and so only one reaction because only one specific substrate fits the active site. Okay. 3. Lipase is an enzyme that digests lipids. Lipids are broken down into smaller molecules. Give one of these molecules. Okay, so fats, okay, just as we break them down with lipase, it's also assisted by bile, which is released from the liver, okay, stored in the gallbladder, and bile comes along and breaks down the fats into small fat molecules which increase surface area. Now, we haven't got to the question yet. It's asking what is fat broken down into. These little bits of fat are broken into two things: glycerol and 3 fatty acids. I think that's the hardest one. Proteins are broken down into amino acids, fats are broken down into glycerol and 3 fatty acids, and starch is broken down into glucose. Okay. Which organ is lipase produced in? Tick 2 boxes. So it's produced in the pancreas. Okay. Definitely in the pancreas, because it's in the pancreas that the small intestine enzymes are made. And then it's also made in the walls of the small intestine. Okay. So that's a quick digestion recap.